Hey, Jared. Here's your feedback for assignment number 5, your materials and methods with the mock data. Okay, so let's start with your title. First things first, overall formatting, remember to justify, left, right, justify all your columns to make sure you're meeting the proper instars format. Other things, though... Yeah, okay, this looks good. I really like how you have the sort of subheadings. Double check the size if that is put out in the author guidelines for this. I feel like the subheading colony rearing should be slightly smaller than materials and methods. Also make sure you have consistent font throughout because that's just a really easy fix. Okay. Now back to your title. So the effects of sterilizing Cochleoma macellaria eggs using formalin and bleach. First things first, great job putting Diptera califordi right after the species name. All you need now is one more parentheses with the author who first, I want to say examined, Lord, who first described macellaria. So put that in there and you'll be fine. Now, the word choice here leaves a little bit of ambiguity. So you want to choose words that are very specific when you're doing a very specific scientific concept. And a lot of these words have a specific definition. Sterilizing is one of those definitions. So to sterilize something, sterile means the lack of living organisms. So when you say you're sterilizing eggs, that means, in the context of entomology, you're making it so those eggs don't hatch. You're making it so those eggs are dead. They have no living organisms in them. I mean, we do a lot of sterilization of eggs or a lot of sterilization of insects so that they only produce non-viable eggs. So reading this title, using that word sterilizing, that's what this implies. Instead, since you want the eggs to be alive, in this particular case, since you're trying to rear out maggots for wound debridement therapy, you want to surface sterilize the eggs or disinfect the eggs. So if you look up disinfecting, that means the removal of microorganisms on a living thing, usually, or on non-living objects. So that would get across what you're trying to do here. And then surface sterilization is also a way you can get that information across. So just be very specific about the types of words that you choose. Otherwise, this is a pretty good title. Make sure you're using title case so all of the nouns and verbs are capitalized. Using formalin and bleach, I mean, that's a very specific ending to the title. So if it had just been the effects of surface sterilizing Cochleoma macellaria eggs using formalin and bleach, that would be a perfect title. Okay, so go on with that. All right. Now, again, I really like how you organized the materials and methods. It makes it very easy to follow, very simple to recreate. There's no confusion. So good job there. Keep that up. All right, now let's start on here. Cochleoma macellaria library harvest from carrion found on the side of the road in College Station, Texas. Perfect. You need to put a comma right after College Station, just the proper way of writing out a city and a state. But otherwise, this is just enough detail so that people who are trying to replicate this know exactly what is happening, but not so much detail that they get bogged down in things they don't care about, like the name of the road. So well done there. Okay, they were then placed on 50 grams of cow liver until they pupated. The beef were placed in a 12 by 12 by 12 fly cage. Okay, that's really, really good. One minor thing, you don't need to capitalize cow here since it's not a proper noun. So that should be lowercase. And then there's no losing of points or anything on this. It's just something I want you to be aware of. Depending on the journal that you go with, you will find that certain editors and journals will ask you to write in a more science-y format, so to speak. So they want you to use as many scientific-esque terms as they can. In STARS, we take pretty much anything because it is a learning process. And then there are other journals that are happy to take these things. But Certain journals, if you were to say something like cow liver, you might get a message from the editor or from the reviewer saying this doesn't sound proper. So they're looking for the more sciencey term there. In this case, it would be bovine liver. This is just something that I've learned to get used to over my years of turning stuff into journals. And I now tend to err on the side of using more scientific-y type language when I'm writing to try to keep from having to revise it, So just be aware that this is perfect for this paper, but keep it in mind in the future as you go on to write for other scientific publications. All right. So good. Adult flies, dry sugar, standard circadian rhythm, schedule. So the standard circadian rhythm schedule, you would need to either define what that means, because this might be different for different authors or for different researchers, depending on their system. Because there's no one standard circadian rhythm for every single organism on Earth. So, an entomologist is going to do something slightly different than a human researcher, who's going to be different than a mammalian researcher, et cetera, and they'll be very different from, say, a microbe or things. So when you do something that is standard like this, make sure you either cite it or define it. So in this case, it was just 12-12 light dark. So it's just as easy to say LD 12-12. On there. I think I have that written out under common mistakes and infrequently asked questions, but if I don't, I will for you. Okay, so that would be a little bit easier to understand there, or at the very least cite where you got that this was a standard circadian rhythm. Okay, after 2 days, remember to spell out words or numbers that are under 10. Inside the fly cage, 50 grams of cow liver was added into the enclosure. Additionally, okay. Okay, so these next two sentences are a little confusing on exactly what you did, because you decided to really spell out what was going on here. You want to make sure that it actually makes sense. So in this case, you could say after 2 days or 48 hours after 48 hours inside the fly cage. First off, that's a little ham-handed the way that you wrote that. You already said they're in the cage. You could just say after 48 hours, 50 grams of liver was presented. And so get rid of sort of like added into the enclosure. That's redundant, because you literally just said fly cage. So after 48 hours, 50 grams of liver was presented to the flies, and this was specifically as a protein meal, right? So you say as a protein meal, and then however long afterwards, an additional 10 grams was added for oviposition. So the way this is written, it sounds like that after 2 days you added 50 grams, and then immediately 2 seconds later added 10 grams. So there's no explanation as to why you did it in two parts or anything like that. So you could simplify that and just say, after 48 hours, adult flies were given a protein meal and then presented with 10 grams of fresh liver as an oviposition medium. Sort of make it very generic like that, where you're saying that you gave it to them, but not down to the exact hours or minutes or whatever. So keep that in mind. Okay, egg preparation and pre-watching. Good. Okay, you got small paintbrush, paper towel. Good. Reverse osmosis water. Perfect. Again, spell out numbers that are under 10. The clutches gently stirred, gently separated, placed in groups of 30 onto filters. Beautiful. 20 micrometers. Yeah. Love it. Make sure that you actually add the manufacturer, city, and state here again. So since they were GE, get the manufacturer, city, and state, so that people can find the same type of filter if necessary. This type of thing is especially important for major bits of equipment in the experiment like this one. I mean, a good portion of the experiment depended upon these filters. So great job putting the diameter size and the micrometer size. But if GE either goes out of business or no longer makes these things, people can still look up what was in these filters. Or if there's something weird going on, they want to know what's in the filters that latched onto some of the proteins or that caused differential filtration of certain substances, So you need to put that manufacturer, city, state there. Same thing with all the rest of this stuff, like the filter holder and the Luer lock syringes, et cetera, Make sure they're all manufacturer, city, state for those reasons. Okay, so eggs are then placed onto a sterilized filter holder. You just said that. So don't repeat things that you've already said. That's just wasting space when you have very little space. So make sure you go through this one more time and don't repeat yourself so often in the writing. Okay. Eggs are placed on a sterilized filter holder. Good. Using the manufacturer instructions. Great. So this is another reason why you need to put the manufacturer, city, state there. If they need to know what those manufacturer instructions are, they should be able to go look them up. If they have the manufacturer, the city, and the state. Done. Okay, 2 milliliters of trip soy broth. Great job defining trip soy broth, TSB. That's the way you do it. Into a sterile syringe. Again, manufacturer, city, state here. Because, especially with this, because they want to know, or people want to know, is this polycarbonate? Was this a different type? Was this glass? What syringe was used? Okay. And then 5 milliliters was sent through the filter. Sent through, the word sent isn't quite right there. Because that implies no direct action by the sender. It's more sort of sent out into the world, and there are other processes there. Since this was forced through the filter as opposed to let sit through gravity, you need to make that distinction. So in this case, we had to use force to get it through this 20 micrometer filter. Otherwise, we could have done a vacuum method where it sort of sucks it through. We could have done just straight gravity. There's a bunch of different ways to get stuff through the filters. By saying sent, that really doesn't give us any information. Eggs were left to soak inside the TSB for 3 minutes. Okay, so that's good. Again, write out numbers under 10. TSB was cereal diluted and plated. What was it plated on? So it was specifically plated on blood agar. Make sure you say that because the medium there is a significant part of this experiment, right? Certain microbes won't grow unless they are on blood agar, which is why we chose this. If you say put them on nutrient agar or another synthetic something, you might get very different results. So always think to yourself, this information, if I don't put it in there, will it possibly change the outcome of the experiment? Okay, the eggs are placed inside cups with Na. Make sure you look up what Na is. This is actually nutrient agar. So that was defined for you, I believe, in the video and in the frequently asked questions and on the common mistakes. So always triple check what these things are. And if you don't know what it is, feel free to reach out to me and I will help you out. Okay, Raymond TSB was enriched by storing at 37 degrees. Good. Perfect. Okay. Although you said remaining and then 100 microliters, you can say, just say 100 microliters of the remaining TSB. That makes it a little more clear as to what exactly you're doing. Good. Now the treatments, eggs were placed on the filter. Again, you've already said that. So if you say it once in sort of a generic thing, you don't need to say it again. You could just say eggs and filter were pre-washed and then placed in, for treatments. So pre-washing, how was any of this different from what you just wrote out in pre-wash? Yeah, so you've already actually written all this out in the pre-wash. That's the whole point of having this previous section. So you can get rid of all this part where it says the eggs were, were placed in the filter and pre-wash and blah, 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 because we know you just said. So keep that in mind. Then the eggs were rinsed in 10 milliliters of insect saline. Excellent. So this is the perfect way of writing out insect saline. You've just given the recipe. So you can, this is probably the best way for this particular thing to write this out. There's another way that you can do this, though, in case you have a really long recipe or something like that. You can, instead of writing out the recipe, you can just cite a paper that has the recipe in it. So just either or. They're both the perfect valid ways of doing this, but just in case for future reference. Now, insect saline is not a product name, nor is it a proper noun, so it doesn't need to be capitalized. Okay, post-egg treatments, good. 10 treatments. Okay. So at this point, you're talking about now the post-egg treatment, or it should be post-wash, right? And so saying post-egg doesn't quite make sense. Like, is this after the egg? So try to be consistent in your labeling and your naming. And then they're rinsed in TSB using all the same steps as pre-wash. That's good. I mean, you could really put those together in egg pre-wash, post-wash to make it a little easier, because this 10 treatments, that's not exactly a post-wash treatment thing, right? That's an actual treatment. So just make sure they're in the correct places. Good job, though, writing out what bleach is. So formalin, formalin is a proper noun, so good job capitalizing that, but it's 5% formaldehyde. So that's what you should have put here, not just 5% formalin, because it'd be like saying, Coca-Cola is 5% Coca-Cola or something like that. That would be weird. So but this is how you should write it out. So that's the name, and then here's the actual scientific information. Bleach, though, is not a proper noun. Unless you were saying Clorox bleach, then it would be capitalized. So be careful there. Okay. Samples are treated in formalin. Good job breaking these up into a sterile syringe. When you start a sentence with a number, you need to spell out that number no matter what it is. So 2.5 should be spelled out. Pass through the filter, allowing the eggs to soak for 5 minutes. Okay. Yeah, that makes sense. After this, the remaining formalin is pushed through. Good. Push through. And rinse with insect saline 2 more times. Okay, the eggs were rinsed again with insect saline. Yeah, okay, because you rinsed it in the pre-wash. Is that right? Okay. Good. Let's see. And then bleach, that looks good. Bleach, 5 milliliters. Cool. Okay. So other than that, that looks pretty good. You did miss one part for the materials and methods. You need to put in there how these data were analyzed. So the scientific, or the statistical calculations that were used. So in this case, it was linear regression, and it was performed on a software package, SPSS. I believe that was the one in the protocol. So you need to add that because, again, if we ask ourselves, if someone did a different statistical test, could that affect the results? Yeah, that's what stats are for, and post hoc tests and all that jazz. And so if somebody doesn't know what type of statistical test that we use, and then they go and try to replicate our results, if they did a different statistical test, they're going to have different p-values and different errors and all that sort of stuff. And then if they use a different post hoc test, they're going to have different things. So you need to say what those are, and then you need to say what software package you used, because you're going to have different limits. You're going to have different cutoffs, all manner of different things in different software packages. For example, if I do a linear regression on Excel, I end up with a different p-value with using the same numbers when I do it on a web browser for some like online web calculator, or if I do it with SPSS, or if I do it with SAS. So that just has to do with the coding and, different scientific notation and stuff. So you need to say what the exact software is and the exact statistical tests are so that you can have somebody else replicate it much more exactingly. That's a word, exactingly. All right. So there we go. Let me know if you have any questions.